Hello everyone. So today I'll be talking about cell division and what are the types of cell division. Okay, especially mitosis and meiosis. So let's start with the class. So first we'll talk about the stages. Okay, so there are basically three stages of cell division. Okay, so first is mitosis, meiosis, and then it's binary fission. So mitosis, what happens in mitosis? There is a formation of identical daughter cells, just like the parent cell. Okay, meiosis stage, it is required for formation of egg and sperm cells. Basically, it's required for sexual reproduction. And last is the binary fission. Binary fission is a type of asexual reproduction wherein what happens when one body it will replicate, okay, to form two bodies, or you can say two parts. Okay, especially it happens in amoeba, then in archaebacteria and all. What happens out there, you can see binary fission. That means binary means what? Two. Fission means to break. So one body, it will break into two parts or two other body. Okay, so here asexual reproduction is occurring. So this is all about three stages of cell division. So now we'll go into the detailed part. So first I'm talking about the mitosis stage. So in mitosis stage, what happens? First of all, as you have seen in my previous class, that mitosis is first observed by, it was first observed by Strasburger in 1875. Okay, and in animal cell it was first observed by W. Fleming or Walter Fleming in 1879. And this person also, he also coined the term mitosis in 1882. Okay, so now what happens in mitosis? Out here, a single cell, it divides into two daughter cells. Okay, and these daughter cells that are formed, they are genetically identical to the parent cell. And mitosis is also a means of asexual reproduction. And what are the stages in mitosis? Prophase, metaphase, anaphase, telophase, and cytokinesis. So there are 5 phases or 5 stages of mitosis. So what are the events that are occurring in mitosis? So as we can see here, I'll explain to you in details. So first of all, we'll talk about the phase. Okay, so we have seen in my previous class, I've shown you about the G1, S and G2 phase. So as we have seen, after G2 phase, it will enter the M phase or mitosis phase. So in M phase, first we'll talk about the prophase. Okay, prophase. So what happens in prophase? Out here you can see that the chromosomes, now first they were all like this. Okay, so what happened out here, these chromosomes, they will condense. Okay, they will condense, or it's written out here, they will condense, and spindle fibers they will start appearing. Okay, also the nuclear envelope wherein this network was present, now these things were present, that nuclear envelope, it will start to disintegrate, it will start to break or disappear. So when that nuclear envelope, it disappears or disintegrates, then those inside those chromosomes, they start to condense or thicken. Okay, so when they will thicken, when they will condense, then it is happening, these are things are happening in prophase stage. Okay, then it will start entering the prometaphase. In prometaphase, what will happen? But as I have said, those things, the spindle fibers, they will start appearing and attaching with the chromosomes that have already been condensed. Okay, so in metaphase, what happens? So in metaphase, basically the chromosomes, they will come and align in the equator, in the center. Okay, so they'll come in the center, and these chromosomes, they are held with the help of spindle fibers. Okay, so they are held with the help of spindle fibers. They are aligning in the center. Okay, and then there comes the anaphase stage. So in what happens in anaphase stage? In anaphase stage, the centromeres, they will divide. Okay, they will divide, and they will the sister chromatids which are present, they will start moving to the opposite poles. Okay, as we have seen, there are two poles out here, one this and this. So in metaphase, they were in the center, but what happens in anaphase, they will start moving towards the opposite side. Okay, so they will start moving towards the opposite side. This happens in the anaphase stage. Okay, and then at the end we come to telophase stage. In telophase, what happens? Once it moves to the opposite side, again this nuclear envelope or nuclear membrane, it will start developing, and these chromosomes, they'll again go in and enter into that envelope. Okay, into the member, and again they will condense. Okay, decondense. That means before what happened, this chromosomes, they had condensed and thickened. Now again they'll go back to the decondensed state. Okay, so they'll again go back to their own own place inside this nuclear membrane, nuclear envelope, not nuclear membrane, sorry, nuclear envelope. Okay, they'll go into this nuclear envelope and they will go and again decondense inside. Okay, and slowly what happens in the next step? Okay, one more thing out here. As you have seen that this they have moved towards the opposite sides, the spindle fibers they'll again disappear. Okay, we have seen in the prophase that spindle fibers they came and they were attaching with the chromosomes, aligning, they were attaching with the centromere of the chromosomes. So now in telophase what is happening? The spindle fibers they are again disappearing. Okay, and next is cytokinesis. So what happens out here? The cytoplasm, this is dividing, and so therefore you can see out here it will divide and they will form their own daughter cells. Okay, then two daughter cells are being formed which can have their own chromosomes. Okay, so out here you can see parent cell, they will again form two daughter cells. Okay, and they will contain identical genetic information similar to the parent cell. That is why it is known as the equational division. Remember, mitosis is equational division. So from basically from 2n, it will again from 2n only. Okay, equational division. So this is about the mitosis stage. Here you can see out here in clearly you can see out here and see in prophase what is happening in prophase. In prophase you can see here this is the chromosomes. They are becoming like condensed and thickening up. Okay, slowly you can see this centrosome out here. Okay, they contain centrioles, so they will start forming the spindle fibers. See, spindle fibers out here, they are formed. You can see spindle fibers out here. Okay, so out here and you can see the chromosomes are starting to align in the center plate, and in metaphase they are completely aligned in the center. Okay, and in anaphase they are being pulled towards the opposite poles, and during telophase and cytokinesis what is happening? Spindle fibers they are disappearing. Oh, see, they are disappearing and a mid body ring is being produced. That is, it is initiating the division of the cell. Okay, it's initiating the division of the entire this cell. Okay, cells. Okay, so now you can see two daughter cells are being formed. Okay, and also the chromosomes they're again going back to decondensed form. Okay, and the spindle fibers they are disappearing. So this is all about the stages of mitosis. Okay, now let's move on to the importance of mitosis. So mitosis is required so that the new cells can grow and the worn out cells they can be replaced. First. Second is that depending upon the kind of cells and species of organisms, the time of meiosis the time for mitosis it will vary. Okay, so it will vary depending on the kind of cells and species of microorganisms and organisms. Also, mitosis it is influenced by the time of day and temperature and chemicals. Okay. Now, next we come to the meiosis part. What is meiosis? So, meiosis was first discovered in sea urchin eggs by the German biologist known as the Oscar Hertwig. Okay, Oscar Hertwig in 1876 he discovered meiosis for the first time in sea urchin eggs. And in chromosomal level, if we talk about in chromosome level, it was again described by Edward van Beneden in Ascaris roundworm eggs. Okay, so basically when we talk about meiosis, we always say that Oscar Hertwig, he's only like we will talk about Oscar Hertwig only as the discoverer of meiosis. Okay, in sea urchin eggs, remember this. And what happens in meiosis? It is a it is necessary for sexual reproduction because it leads to production of sperms and egg cells. Okay, so out here what is happening? Reduction division, because here what happens? It leads to formation of 4 sex cells. Okay, so one germ cell divides into 4 sex cells. Okay, it may be either egg or sperm, and each will have the half the number of the chromosome of the parent cell. So what happens in meiosis? See, meiosis is similar to mitosis, but there are some basic differences. Like in meiosis, what happens in prophase stage? Okay, in prophase stage, see here also you can see when interphase when it enters the prophase stage, you can see out here the chromosomes they are again becoming condensed, and spindle fibers they are occurring in prophase. Out here you can see out here, so basically there are 2 chromosomes. Okay, two chromosomes are there, and 4 chromatids are there. Okay, so here what happens, what is happening in the initial stage, okay, 4 chromatids and 2 chromosomes are there. And here you can see in outer what happens, what is happening, the chromosomes, they will exchange the information by the process known as crossing over. Okay, so after crossing over occurs, it's a bit complicated than the mitosis stage. Here recombination is occurring, crossing over is taking place, okay, in prophase 1. And in metaphase 1 again same thing. In metaphase it will slowly align in the center in the equatorial plate, and anaphase again they are pulled back. Okay, in metaphase what happens, happening in metaphase, this there is a process known as independent assortment. Okay, so what has happening from the parent cell, one is one, you know that chromosomes there, some is like half is coming from the parental and half from the, okay, it's being formed, right, its replication is occurring new, and it's being formed from one from template and one the new one. So in metaphase what is happening, independent assortment is occurring, wherein selection is being taken, it's selected like which chromosome will go to which information, genetic information will go to which chromosome. That way it's like it's differentiating. Okay, independent assortment is occurring in metaphase out here. Okay, then anaphase 1, out here it's being pulled towards the poles, and in telophase 1 division is occurring. And cytokinesis, you can see the last one, cytokinesis in meiosis 1. Here what's happening, it is leading to formation of two chromatids and 1 chromosome. Okay, here remember 2 chromatids and 1 chromosome. Okay, so remember this. Okay, here it was 4 chromatids and 1 chromosome. 4 chromatids and 2 chromosomes, and here it is dividing. Okay, it's reducing, it's forming 1 chromosome to 2 chromatids. Okay, each in for each chromosome. Okay, out here. Now again in meiosis 2 is occurring, wherein again this is again dividing mode. It's dividing to form out here. Okay, it's dividing to form out here 4, you can see out here, see 4 haploid cells. Okay, 4n. Okay, 4 haploid cells, not 4n, sorry, n. Okay, it's forming 4 daughter cells. Okay, and each having haploid number of it's known as haploid number of chromosomes. Okay, so out here 4, you can see 4 is out here formation, and with n number of chromosomes, or you can say haploid. Okay, so this is happening in the meiosis 2. Okay, so yeah, so here what we're getting, okay, for here it was 4 chromatids, 2 chromosome. Here there is 2 chromatids, 1 chromosome, and here it is 1. Okay, 1 chromatid per cell. Okay, out here see n. Okay, so this is all occurring in the, I'll tell you in detail. Basically remember this, they're forming 4 daughter cells. Okay, and it's reducing. Okay, it's reducing. Now in importance of meiosis, see, it's all, I've already told you, it's a means of sexual reproduction. It leads to formation of gametes, or you can say sex cells. Okay, and it maintains the constant number of chromosomes by reductional division, and is responsible for the diversity among species. This process is a bit complicated, therefore it's a slow process. Okay, so here you can see out here, see this, I was talking about this step. So here you can see in mitosis what's happening. See, parent cell is replicating, it's forming 2n. Okay, and then 2n is again dividing and forming 2n. So basically from 2n you are getting 2n. So it's same equational division. But here you can see it's starting with 2n. Okay, it's again replicating, again forming 2n. Okay, but then again when again division occurs, then they're forming n. This is what I was telling you in my previous slide, that 4 daughter cells are being formed with n number of chromosomes. Okay, n. Understood this? So remember 2n is reducing to n. Okay, out like that, and out here 2n to 2n. Therefore this is known as equational division, and this is known as the reductional division. So what are the differences between mitosis and meiosis? Mitosis is occurring in somatic cells, and meiosis in germ cells. Okay, and mitosis, it leads to the cellular growth, cellular proliferation, and meiosis it leads to the sexual reproduction. In mitosis there are 2 diploid daughter cells are being formed, but in meiosis 4 haploid daughter cells. I told you already, 4 haploid daughter cells are being formed. Okay, outer chromosome number remains the same, but outer chromosome number, they have become half. Okay, so if it's 46 in the parent, it will become 23 in the daughter cell. Okay, Out here in mitosis, no crossing over takes place, but in meiosis, crossing over is taking place. Now, in mitosis, if there's a malfunctioning occurring, it will lead to cancer. But in meiosis, since it's related to germ cells, it will lead to genetic disorders. Okay, and at the last, mitosis, it is out here equational division is occurring, and in meiosis, reduction division is occurring. So this is all about mitosis and meiosis. I hope you have liked the class. If you have liked it, please press on the like icon, share it with your friends, and subscribe to my channel for more such videos. Thank you so much for attending today's class.